Osmania University, Hyderabad. Professor Pawar earned her PhD from Osmania University, Hyderabad. She is also the recipient of Young Scientist Award by PMMRC, as well as a DST Voice Cast Fellowship. Professor Smita is also an excellent teacher, as she is a recipient of a State Teacher Award. I would like to request Professor Smita to speak.
And thank you for that excellent introduction. On the outside, let me thank the organizers for having me here. It's so wonderful being part of this great conference. Secondly, after such experience, people have talked here, so I'm a bit shaky. But they're putting up so much of experience behind them. So I'll try not to get down the tempo what they've built up. So this is, as rightfully as introduced, so this is Professor Smita Pawar, Head Department of Genetics and Biotechnology from Osmania University. So before I could start this topic, I'll just try to briefly describe how I fit into this. I'm basically a cancer biologist, which working on genomics part of it, and why cervical cancer. Cervical cancer, when I started, I wanted to work in cancer biology, but when I was asking myself, like, what cancer should I be working on? Why did I pick up this? Very first reason, if you see, breast cancer happens to be the most common cancer, and then I think CC is coming up in a very big way, and I think it is not very long that it's going to take over cervical cancer. Secondly, it gives you a very broad window for you to work with. There are many cancers that you really do not have the chance to help the patient, but here, one thing that is possible is first, by identifying it at a very early stage, we can reverse it. Secondly, it gives you a lot of time for you to treat the patient. Almost you get a window of 10 years, which most of the cancers do not give you. So these are the two reasons why I started with this. And then how did I start, this work start off? We have done this genomic study where the data regarding Indian population in specific is very, very scarce. So we did a very small pilot study with the South Indian, especially from one cancer hospital, that is MNG Cancer Hospital in Hyderabad, which caters to all of the Telangana. So the whole state's cancer patients who are like below the poverty line are sent to this hospital because that is the only hospital that caters to it. So when I've seen the scenario there, till date I think there are hardly 2 or 3 samples that I've got which are HPV negative, but most of them are HPV positive. So I can say that from my sample, 99% of the samples are HPV positive. That was the first thing, and then the data regarding the population, because the socio-economic status also plays a very crucial role in cervical cancer. So I just wanted to see how it is it really different? No, it's not supposed to be different, but yes, what is the finding that I get from my study? From the transcriptome data that I got, the summary of it, I could say that the genes are the same which are playing a role, but yes, the ranking is changing. The most prominent gene in the Caucasian population is not seen in us, and the ones which are playing prominent role here are not really figuring in the top 10 or the top 20 or top 100 genes if you want to take. So that is how this whole study started off with. Then we identified certain genes which were playing very crucial role, but we are not identified by the other population, definitely not in cervical cancer. One such gene is osteopoietin. Gopal Kunde sir is here, who is an expert in this field. And then when we started looking at what are the other ways and methods of stopping this gene or inhibiting this gene, the expression was not only seen in the cancer patient but also in the recurrent cases. We had done a transcriptome analysis for the recurrent cases where this gene was constant in both the population. Though the sample size was not really huge, but it showed that even for the recurrence, it was playing a very major role. So we had picked up that gene, and then we started looking for, are there any known analogs? Rather than starting to develop a new molecule which is not our expertise, we wanted to see which is already available. And for our good luck, you can say that andrographite happened to be one of the biggest hits, which shows really good compatibility in inhibiting in silico with SPP1. So here, secondary metabolites as adjuvants for cervical cancer, a multi-target therapeutic approach. So the whole idea of this project is, cisplatin happens to be the only drug that has been administered to the patients who are coming to this hospital. the resistance definitely is there. So we have conceptualized this idea because some of the genes that have come up. So if we can add something with cisplatin, which can make it more effective. It's not like replacing it, yes, something which will make it more effective. So we started with this idea. So as I just generally, this is the general introduction, I think, which I will skip here. So here, cisplatin is the drug treatment used as chemotherapeutic agent for many years and in the later stages of the cancer. As I've said you, one thing is HPV positivity, and secondly, most of the samples that we are getting are only at the stage 2A, 2B or 3. That is, in the first stage we hardly have any samples to work with. So I will come to this picture once I finished the whole thing. So we started with the set of genes that we have identified in the study, where PI3K was the biggest hit, NFKB, COX2 and PTEN. So the same data we had to analyze because our sample size was very less. We have analyzed it across the other databases that were available, and yes, it was showing the same kind of a pattern across various populations. Now the same thing we have reconfirmed it with histochemical analysis, IHC. These are the patient samples. If you look at it, the PI3K, you have a very strong positive. Then the same thing with the NF-kappa B, where we see a very strong positivity here when compared to the controls. The COX also has a very strong cytoplasmic presence, and the PTEN, we hardly find any PTEN in this. The data was further analyzed by doing the qPCR analysis, where the similar results were seen. Further evaluated by the ROC curves. And these are the I'm not put most of the pictures here, just a representative picture where we have done the molecular docking, and these docking studies have been done at VIT Vellore, where Dr. Anthony is our collaborator who does all the in silico studies, and he gives us the list of the molecules. So andrographlite was one of the biggest hits that was proposed by the VIT lab. Then we validated this, whether, there is already a lot of literature that is present. Still we had to redo this thing again on HeLa and SIHA cells, and here we validated the inhibitory effect of andrographlite on the cervical cancer cells. As you see, it does show good inhibition here, and this paper was published in 2021. So this is the same thing showing the inhibitory effect of the andrographlite. We perform certain set of assays as a cancer biologist, as we are all well aware of the transfield migration assay. We are just checking whether the andrographlite is able to inhibit this migration. When you see that the HeLa is treated with andrographlite, there is a gradual reduction in the migration as well as in the SIHA cells. Then the matrogen invasion assays, as the concentration is increased from 5 micromolar to 10 micromolar, we find that the migration of the cells has drastically decreased, both in HeLa as well as the SIHA cells. Now looking at what are the targets that this andrographlite is hitting. As I told you, PI3K was the biggest hit in the transcriptome data that we had generated from the patient samples. So we started with the PI3K, and here you can see that the PI3K upon the treatment, gradually you can see a decrease, a substantial decrease in the PI3K, and then PAKT also has gradually decreased, but no difference was observed with the AKT. The same kind of results we have seen with the SIHA cells. The similar experiments were performed with the NF-kappa B, COX2 and PTEN. We find substantial increase of the PTEN with the treatment of andrographlite, and the COX2 and the NFKB, we see it gradually going down. The same results we have seen both in HeLa as well as in SIHA cells. We looked at some of the apoptotic markers. The BAX has gone up and the BCL has gradually come down. Similar results again are repeated in the SIHA cells. We did an ICC assay to see the NF-kappa B. So this is nothing when compared to the slides that I have just seen. So here also what we see is the NF-kappa B is gradually being decreased with the treatment of the andrographlite. So we try to replicate these experiments in vivo this was a bit of a difficult task for us because maybe the model that I had used was not an appropriate model, but because we had started with C57BL6 BL, we had taken these mice, but initially when we tried to grow the tumor with the HeLa cells, we couldn't do it. So then we had to suppress the mice, and then we had to go ahead with the treatment. That gave us certain, the results were seen, but I think this is the protocol that we have standardized, and I think it's a standard protocol done by most of the labs. If you look at the tumor size, when compared to the control, the treatment have shown gradual decrease in the tumor size. The ILCs were performed with the tumor tissues that were obtained from the animal. So in the first one, you find that the PI3K, the level is mild to no IHC that is being seen. NF-kappa B is also mild to moderate, and the COX-2 levels also have decreased from mild to moderate. And P10 level has predominantly seen in the nucleus and from no reactivity to moderate level. So this is in sync with what all data that had shown you in vitro. So in vivo also andrographolite was showing the same kind of an impact. So this is what the cartoon that first I skipped. So here andrographolite is not only targeting one, but many hits are there. So it is very effectively inhibiting the PI3K, NF-kappa B, COX-2, and at the same time it is initiating or there's an upregulation of the apoptotic markers. So this is what we are proposing is a combination therapy of for treatment of cervical cancer, that is andrographolite in combination with cisplatin. So we are now developing cisplatin resistant lines to see whether andrographolite can make any difference to that. So that is still the study, that is still going on, and this project has been funded by SIR. So thank you, and I'll be able to take some questions.
Professor Smita is a nice talk. So just quick 2-3 questions and comments.
Yes.
Is andrographolite working HPV positive cervical cancer or HPV negative cancer?
So right now we have shown only the HPV positive, so negativity.
Second question.
Yeah.
When you talk about PI3 kinase, it has two subunits. One is catalytic, another inhibitor.
Yes.
P110 and P85.
Yes.
You have shown P85, expression of P85 going down.
Yes.
You should check the level of phosphorylation of P110 and P85.
Sure.
Third question, you shown that NF-kappa B. NF-kappa B main subunit is P65. Whether heterodimers or homodimers along with P50 or P65, P65. You should check the level of phosphorylation of P65 rather than saying only NF-kappa B.
Sure.
And what is going on? Is andrographolite enhancing or inhibiting P10 expression? What is going on?
So
or
P10 expression.
of P10?
So right now we have looked at the P10 expression only, and it has shown increase in the expression of the P10 levels, both in vitro and in vivo.
Because being a tumor suppressor, maybe you should have a combination of P10 along with andrographolite. That might be other than that other chemotherapeutic drugs.
Thanks for this suggestion, so definitely we'll try to incorporate, because still this is an ongoing project.
Thank you.
Any other questions? Thank you. I would again check I would again request Professor Ranjan to
Thank you.